Hello everyone, I am Nandini Marappan from the PG and Research Department of Microbiology, Vivekananda Arts and Science College for Women, Sangagiri, Salem. I am going to present the topic from Soil and Environmental Microbiology, Plant Growth Promoting Rhizobacteria, with a focus on non-symbiotic microorganisms such as Azotobacter and Azospirillum, under the guidance of Dr. R. Dinesh Kumar, Assistant Professor from the PG and Research Department of Microbiology, VIAS. We will understand what PGPR are, how non-symbiotic bacteria function in the soil, their mechanisms of promoting plant growth, and their applications in sustainable agriculture. Let us begin the presentation. Here is the outline of today's presentation. First, we will start with an introduction to PGPR. Then, we will look at their classification, characteristics, and mechanisms of action, including both direct and indirect plant growth promotion. Next, we will discuss the role and importance of PGPR in agriculture. After that, we will focus on two important genera, Azotobacter and Azospirillum, covering their morphology, characteristics, uses, and mode of action. Finally, we will compare the differences between Azotobacter and Azospirillum. Let us begin with the introduction. The term plant growth promoting bacteria refers to beneficial microbes that colonize the roots of plants, also known as the rhizosphere. These bacteria help in enhancing plant growth by improving nutrient availability and producing useful compounds. The concept of plant growth promoting rhizobacteria or PGPR was first introduced by Cloper and Stroth in the year 1981. Now, let us move on to the classification of plant growth promoting rhizobacteria. PGPR are classified into two main groups. First, ePGPR and second, iPGPR. The extracellular PGPR live in the rhizosphere on the root surface or in the spaces between root cortex cells. Examples of ePGPR include Agrobacterium, Azotobacter, Azospirillum, Bacillus, Pseudomonas, and Serratia. On the other hand, the intracellular PGPR are generally found inside specialized nodular structure of root cells. They belong to the family Rhizobiaceae, which includes Bradyrhizobium, Mesorhizobium, and Rhizobium. Now, let us look at the main characteristics of PGPR. They must be able to efficiently colonize the root surface. Next, they promote plant growth by providing nutrients and producing growth hormones. And they should be able to survive, multiply, and compare with other microorganisms in the soil at least long enough to express their plant growth promoting and productive functions. Finally, PGPR also play an important role in the biological control of plant pathogens. Now, let us understand the mechanism of PGPR or the way they promote plant growth. The action of PGPR can be explained in two ways, one direct promotion and indirect promotion. In direct plant growth promotion, PGPR fix atmospheric nitrogen, solubilize phosphates, produce important plant hormones such as auxin and gibberellins, and also produce siderophores that help in nutrient uptake. In indirect plant growth promotion, PGPR protect plants against diseases by producing lytic enzymes and antibiotics. They also induce systemic resistance in plants and produce exopolysaccharides, which improve root production and soil structure. So, PGPR not only supply nutrients but also protect plants from harmful microorganisms. Next, let us see the role of plant growth promoting rhizobacteria in nitrogen fixation. This picture shows how PGPR interact with plant roots. PGPR colonize the root surface and release phytohormones, lytic enzymes, and secondary metabolites. These compounds help in root hair formation, lateral root development, and primary root elongation. Next, PGPR also contribute to nutrient supply by fixing nitrogen, solubilizing phosphorus, and producing siderophores that improve iron availability. Inside the root cells, PGPR influence gene transcription and metabolite biosynthesis, which supports better root growth and plant health. Overall, PGPR enhanced plant nutrition and root architecture, leading to stronger and more productive plants. Let us discuss the direct mechanism of plant growth promoting rhizobacteria, specifically nitrogen fixation. Nitrogen is one of the most essential nutrients required for plant growth and productivity. Even though our atmosphere contains about 78% nitrogen gas, it is in an inert form and cannot be directly used by plants. Here, biological nitrogen fixation carried out by microbes such as PGPR plays a crucial role. These bacteria convert atmospheric nitrogen into plant usable forms like ammonia. In fact, biological nitrogen fixation accounts for nearly two-thirds of the total nitrogen fixed globally. The process of nitrogen fixation is carried out by a special enzyme known as the nitrogenase complex. This enzyme is highly complex and plays a key role in converting atmospheric nitrogen into a usable form for plants. According to Dean and Jacobson in 1992, nitrogenase is a two-component metalloenzyme. The picture here shows the nodulation process. The rhizobium bacteria come in contact with the root hair. This interaction causes the root hair to curl, and then an infection thread is formed. Through this thread, the bacteria enter the root and initiate the formation of nodules, where nitrogen fixation finally takes place. Nitrogen fixing organisms are generally divided into two categories. The first group is symbiotic bacteria, such as members of the family Rhizobiaceae, which form associations with leguminous plants like Rhizobia, and also with some non-leguminous trees, for example, Frantia. The second group is non-symbiotic nitrogen fixers. These include free-living, associative, and endobiotic microorganisms such as cyanobacteria, as well as Azospirillum and Azotobacter. These organisms play a vital role in maintaining soil fertility by naturally enriching nitrogen levels. Phosphorus is abundantly present in soils, but most of it exists in insoluble forms, making it unavailable to plants. Plants can absorb phosphorus only in two soluble forms, the monobasic ion, like dihydrogen phosphate ions, and the dibasic ion, like hypophosphorus acid. Microorganisms with phosphate solubilizing activity, commonly known as phosphate solubilizing microorganisms or PSM, play a vital role in converting insoluble phosphorus into these available forms. This natural process provides plants with essential phosphorus and can serve as a sustainable alternative to chemical phosphate fertilizers. Let's see the role of bacteria in phosphorus mobilization and immobilization in soil. Several bacterial genera such as Azotobacter, Bacillus, Beijeringia, Pseudomonas, Rhizobium, and Serratia are considered the most important phosphate solubilizing bacteria. As we can see in the picture, soil phosphorus exists in both inorganic and organic forms. Through the processes of mineralization and solubilization, bacteria like Rhizobium, Bacillus, Pseudomonas, and Enterobacter convert these forms into bioavailable phosphorus for plant uptake. On the other hand, bacteria such as Azospirillum and Azotobacter are involved in immobilization, which temporarily locks phosphorus in microbial biomass. Together, these processes maintain the phosphorus cycle and ensure its availability to plants. The role of phytohormone production in plant microbe interactions. Phytohormones are organic compounds other than essential nutrients that regulate germination, growth, metabolism, and various physiological activities in plants. Among them, indole 3-acidic acid plays a crucial role in rhizobacteria plant interactions by promoting root development and overall plant growth. In addition, IAA is also linked with plant defense mechanisms, helping plants respond to phytopathogenic bacteria. Interestingly, studies show that the external application of IAA can influence plant susceptibility to bacterial pathogens, which demonstrates its dual role in both growth regulation and defense. The picture explains how stress controlling bacteria help plants by reducing ethylene levels in the root system. Normally in plants, the amino acid methionine is converted to S-adenosyl methionine, which is further converted into ACC, or 1-amino-cyclopropone 1-carboxylate. ACC is then broken down by ACC oxidase into ethylene. Ethylene plays a dual role. While low levels support root elongation, high levels are produced under stress conditions and can inhibit plant growth. Certain beneficial bacteria produce an enzyme called ACC deaminase. This enzyme breaks down ACC into ammonia and 2-oxobutonate, thereby reducing the amount of ACC available for ethylene production. As a result, the ethylene level in the plant root decreases, helping the plant to minimize stress responses and continue normal growth and root development. Next, sideropore production by microorganisms. To meet their iron requirements, microbes produce special low molecular weight compounds called sideropores, which act as iron chelators. These sideropores are secreted into the environment where they bind and solubilize iron, forming a ferric sideropore complex. This complex can then diffuse back to the microbial cell surface for iron uptake. In soil, sideropore production is very important because it determines how effectively microorganisms can acquire iron and in turn influence plant growth and development. Moreover, microbial sideropores also benefit plants since certain plants are able to recognize the ferric sideropore complex and utilize it for their own iron nutrition. This enhances plant health and supports overall development under iron limiting conditions. Next, the indirect mechanisms by which plant growth promoting rhizobacteria support plant health. First, lytic enzymes. Certain rhizobacterial strains can produce enzymes such as chitinases, dehydrogenases, lipases, and proteases. These enzymes break down the cell walls of pathogenic fungi and other harmful microorganisms, thereby protecting the plant. Second, antibiotic production. Many rhizobacteria produce antibiotics, which is considered one of the most powerful and well studied biocontrol strategies. These antibiotics suppress phytopathogens and prevent diseases, ultimately promoting plant growth. Examples of such antibiotics include tropolone and penazine. Through these indirect mechanisms, rhizobacteria help plants fight pathogens and maintain a healthy growth environment. Next, the concept of induced systemic resistance. Induced systemic resistance is a plant defense mechanism that is triggered by signaling molecules such as jasmonate and ethylene. These hormones activate the host plant's immune system, making it more resistant to a wide variety of plant pathogens. In simple, ISR can be defined as a physiological state in which the plant's defensive capacity is enhanced in response to specific environmental stimuli. As a result, the plant's natural defenses are strengthened, allowing it to respond more effectively to future biotic challenges. Thus, induced systemic resistance act as a form of priming, preparing the plant to better withstand attacks from pathogens. Next, the production of exopolysaccharides plays an important role in biofilm formation. This process influences how microbes interact with plant roots. Exopolysaccharide producing microbes have several crucial functions. They provide protection from drying, help plants resist stress, allow microbes to attach to surfaces, and also strengthen the plant's defense system against invasion. In addition, when these microbes colonize plant roots effectively, they make phosphorus more available by converting it from the insoluble form in the soil. Finally, they help in circulating essential nutrients to the plant, ensuring proper growth and development while also protecting the plant from harmful pathogens. Next, let us see the role of plant growth promoting bacteria. They help plants tolerate abiotic stress conditions such as drought, salinity, or temperature extremes. They are involved in the production of volatile organic compounds which support plant health and growth. They also improve nutrient availability in the soil, making it easier for plants to absorb essential elements. In addition, these microbes act as plant growth regulators, promoting overall development. Finally, they produce productive enzymes such as chitinase, glucanase, and ACC deaminase. These enzymes play an important role in preventing plant diseases and ensuring healthy growth. Next, let us understand the importance of plant growth promoting rhizobacteria. PGPR are highly beneficial for plant growth and development. In today's world, where there is a growing international concern for food security and environmental quality, the use of PGPR in agriculture has become very important. One of the main reason is that they help reduce the dependency on chemical inputs. They also play a vital role in recycling soil nutrients, ensuring that the soil remains healthy and productive. Towards achieving sustainable agriculture, crops supported by PGPR can gain several advantages such as improved disease resistance, tolerance to salinity, trough, and heavy metal stress, as well as enhanced nutritional value. Next, the morphology of Azotobacter. Azotobacter are motile bacteria, usually oval or spherical in shape. They are gram-negative and heterotrophic and are naturally occurring nitrogen-fixing bacteria. These organisms are commonly found in neutral or alkaline soil. They are free-living, aerobic, and can form cyst with a productive capsule. Example, Azotobacter crocacum and Azotobacter baegiensis. Overall, Azotobacter plays a very important role in agriculture as they help plants in fixing atmospheric nitrogen, thereby improving soil fertility and supporting plant growth. Next, discuss the role of Azotobacter. Azotobacter plays a key role in nitrogen fixation. It converts atmospheric nitrogen into ammonia, which can be easily utilized by plants. It is commonly found in the rhizosphere of several crops, including rice, maize, sugarcane, and vegetables. When seeds are inoculated with Azotobacter, it enhances the uptake of essential nutrients such as nitrogen and phosphorus, particularly in crops like wheat. Overall, Azotobacter has a great potential to improve plant nutrition, soil fertility, and crop productivity in a sustainable way. Next, the characteristics of Azotobacter. Azotobacter is a highly efficient in promoting plant health and growth. It has a rod-shaped structure and belongs to the group of gram-negative bacteria. This bacterium is motile because it has a flagellum which helps in movement. One of its unique features is the ability to survive under harsh environmental conditions by forming cysts. This adaptation allows Azotobacter to remain active and continue supporting plant health even in unfavorable environments. Next, the important uses of Azotobacter. Azotobacter as a biofertilizer plays a vital role in ensuring plant health. It is widely applied in agriculture to improve soil fertility and stimulate plant growth. Its presence is highly significant in the formulation of biofertilizers. Beyond agriculture, Azotobacter is also crucial in the production of alginic acid, which is well known in medicine for its application as an antacid. Additionally, it is important to note that Azotobacter has applications in the food industry as well, making it a microorganism of great industrial importance. Next, the morphology of Azospirillum. Azospirillum is known as a plant growth promoting diazotroph. It is a free living gram negative bacterium. These bacteria are rod shaped and do not produce spores. They are commonly found in freshwater environments and in close association with plant roots in soil habitats. Azospirillum is microaerophilic, meaning it is aerobic in nature but can also survive under low oxygen conditions. This unique ability helps it adapt to different soil environments. The picture showing the role of biostimulants. In this diagram, we can see how Azospirillum plays an important role in supporting plant growth under stress conditions. At the root level, Azospirillum helps in producing phytohormones, exopolysaccharides, and ACC deaminase, which together promote root growth. This improves nutrient uptake and helps plants tolerate salinity stress. by increasing potassium and calcium absorption. In the upper parts of the plant, Azospirillum also reduces oxidative stress by activating antioxidant enzymes which protect the plant during both abiotic and biotic stress. Additionally, it enhances induced systemic resistance, providing protection against phytopathogens. It also promotes plant growth through osmotic adjustment and by stimulating the synthesis of protective proteins. Overall, this picture highlights how Azospirillum functions as a biostimulant, helping plants survive under oxidative stress, water stress, and salinity stress while also improving their overall growth and resistance. Now, the characteristics of Azospirillum. These microbes secrete plant hormones that help the roots grow more branches and root hairs, which enhances nutrient absorption. They also make essential nutrients like nitrogen, phosphorus more available to the plants. In addition, they produce antioxidants that protect the plants during stressful conditions. Another is the important point is that they compete with harmful microbes, which makes the plant less susceptible to diseases. So overall, these characteristics play a key role in improving plant health and productivity. Next, let us see mode of action by azospirillum. They help in increasing photosynthetic pigments, which improves the plant's ability to produce food. They also enhance seed germination and play a key role in nitrogen fixation, making nitrogen available to the plants. Another important action is that they increase the permeability of the plasma membrane, which improves the uptake of essential nutrients. At the same time, they produce antimicrobial and antifungal compounds that protect plants from harmful pathogens. On the other side, they improve plant tolerance by introducing systemic resistance, which helps plants with stand trough and water stress. They also secrete important plant growth hormones such as IAA, gibberellin, and cytokinin, all of which regulate growth and development. Additionally, they can reduce the soil pH through phosphorus solubilization, making phosphorus more available to the plant. They also produce siderophore, which bind iron and make it unavailable to disease-causing microbes. So overall, through these combined actions, the microbes significantly enhance plant growth, stress resistance, and protection against diseases. Next, the difference between Azotobacter and Azospirillum. Azotobacter is an aerobic soil-dwelling bacterium, whereas Azospirillum is microaerophilic and usually colonize plant surfaces. In terms of oxygen requirement, Azotobacter needs high oxygen concentrations to function, while Azospirillum can also survive in low oxygen conditions. When we look at their shape, Azotobacter cells are oval or spherical, but Azospirillum has an oblong, rod-shaped structure. Regarding cyst formation, Azotobacter forms cyst to protect itself under harsh environments, but Azospirillum does not form cysts. Taking about their functions, Azotobacter mainly act as a nitrogen fixer and also help in producing biofertilizers and biopolymers. And the other Azospirillum is mainly useful as a plant growth promoting bacterium. They also belong to different families. Azotobacter is part of the Pseudomonaceae family, while Azospirillum belongs to the Azospirillum family. Finally, the temperature range for Azotobacter is between 20 to 30 degrees Celsius, whereas Azospirillum can tolerate a wider range from 5 to 42 degrees Celsius. Thank you so much for your time and attention.